Okay, so this part I'm going to start at what would be section 8.4, and this has to do with sexual reproduction. So the last video was asexual, and then this one's going to be sexual. So when we talk about asexual, would be like this bacterial cell splitting into two identical bacterial cells, whereas sexual would be this butterfly mating with another butterfly, laying eggs, and then the eggs hatching, and we're actually showing complete metamorphosis there. And we all know the whole story. The eggs go into larvae into caterpillars, then caterpillars form the chrysalis, and then they hatch back into fully mature butterflies. All right, the thing with asexual is that it is very fast, but the offspring are identical. With multicellular organisms, we usually bud or we lay asexual eggs. Single cellular, we use a term called binary fission. For sexual reproduction, we have to have fertilization. So we're going to have an egg and a sperm, and they come together, and they're going to make genetically unique offspring, which may be better able to survive the environment or any changes in the environment. So in order to go from having 46 chromosomes in a diploid cell, meaning we have two copies, we have one copy from mom, one from dad, into an egg or a sperm that has only one copy, which is called haploid, one N, we're going to undergo a process called meiosis. And so this is the only place that meiosis takes place, and that is to make gametes. Gametes is the technical name for eggs and sperm. We're going to fertilize that egg. That fertilized egg is called a zygote. Gametes are haploid. Zygotes are diploid. And so they are going to have two sets of homologous chromosomes, meaning they have one copy from mom, one from dad. Your somatic cells are body cells. They are all diploid. The only cells in your body that are haploid are your gametes. So if you are a woman, your eggs. If you are a man, your sperm. So again, this is only going to happen in the testes and the ovaries. They're going to undergo meiosis. The female is going to make eggs. The male will make sperm. We fertilize. We go back from being 1N to 2N. There's our zygote. You can see mom and dad's both copies in fertilization. And now we're going to undergo mitosis. That one fertilized egg, that zygote, will then divide by mitosis to make that multicellular organism. So chromosomes, we talk about this term homologous chromosomes. Those are chromosomes that have the same type of information on them. So like mom's chromosome 1 and dad's chromosome 1 will have the same info on them. Now they may not have the same exact version of that gene, like maybe dad's tall, mom's short, but height will be the same thing on that, will be on the same chromosome. So we know that humans have 22 pairs of homologous chromosome. We call those the autosomes. So chromosomes 1 through 22 have no role in sex determination. The allosomes, or the sex chromosomes, the X and Ys, those are the chromosomes responsible for determining whether you are genetically a male or a female. Phenotypic males have an X and a Y, and our phenotypic females. Phenotypic means like, what do we present as? Which genitalia do we make? We'll talk about the difference between geno and phenotype when we get to our genetics. So this word's kind of prematurely put in here, but basically it will present itself as male. If you're X and Y, you will present yourself as female if you have two X's. We are not referring to gender. We are talking about sex here. So I don't want to get into that debate. Basically, do you have the male parts or the female parts? Okay. Without getting a little too specific here. So we have our human karyotype. This is a picture that shows us our chromosome pairs 1 through 22 in humans, and then our allosomes are our sex chromosomes. This would be an X and a Y. So this individual would be male. So when we talk about meiosis it's going to make our gametes diploid. It's going to take our diploid cell and make it haploid. There are two complete sets of cell division. And so we are going to refer to one as meiosis 1, and the second set is meiosis 2. We are also going to have, this is still part of the cell cycle. So we're still going to have we're going to have that interphase where we have to go through G1 acid and G2. So we have our gap 1 where we are going to have that normal functions of our cells. We're going to be transcribing, translating. We're going to learn about that in chapter 10 and undergoing cellular respiration. And then we're going to be, if we're given the signal, we'll go into synthesis where we're going to make our sister chromatid. And then we'll go into G2 where we're going to prepare for cell division. In this case, instead of it being mitosis, we'll be going into meiosis. So again, prophase 1 is very similar to mitosis. The DNA is going to wind all up. The nuclear membrane is going to break down, with the exception of the homologous chromosomes are going to find each other. So dad's chromosome 1 will find mom's chromosome 1. And they're going to hang out together in what's called a tetrad. Tetrad because there are 4 copies, right? 1, 2, 3, 4. So there are 4 copies of all genes sitting in that one location. So we call it a tetrad. Now, what may happen is these homologous chromosomes may get a little too close. And so we could have what's called a crossing over. So a crossing over event is one way of getting genetic variation. And so we have what is called a chiasmata, and the chiasmata is where those two, it's like kind of like in this picture here, that would be our chiasmata here and here where they're held together. And so at that chiasmata, we may have a crossing over. So notice we have swapped our arms, our sisters here of our tetrad. And this again can lead to new genetic variation. Very important. We need genetic variation. That is important for the success of our species to be genetically diverse. Now we kind of divide, we have our PMAT. We're going to talk about prometaphase. Prophase takes so long. There's so many things going on in prophase. So we kind of have it broken into prophase and then kind of like almost to metaphase. So that's prometaphase 1. And in prometaphase 1, we have these new combinations of our homologous chromosomes, and they're going to slowly pair up. Like I said, the nuclear envelope is fully dissolved, and we're going to pair up across from each other. Okay, not single file. Single file is mitosis. We're going to pair up with our partners across from each other for metaphase. So we're going to line up on that metaphase plate with our homologous partner across from us. Now, there are 2 ways that we could set this up. Okay, now we can set this up so that we have, notice all the blue are on one side and all the red are on one side. So on our metaphase plate, we can have all dads on one side. And so when it splits, I call this a divorce. You split your parents up. You have one gamete going to have all moms and one that's going to have all dads. But there's no specific way that these chromosomes have to line up on the metaphase plate. They can also be reversed. So notice how here on my metaphase plate, I have dads and moms on the same side. So when I split up, I have a combination of mom and dad. So this is referred to as the law of independent assortment. And so think about it. When you have 23 pairs, you could have mom, dad, mom, dad, mom, dad, 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 dad. Or you could have all mom on one side, all dad on one side. It is completely random. So they will independently assort as long as they're paired up. And this is our second source of genetic variation. So once we end up lining up our parents, we're going to have the divorce. We're going to split them apart in anaphase 1. So we split our parents apart, and this is where we're going from diploid to haploid. And so next we're going to go into telophase 1 and cytokinesis. This is, like I said, these steps are very similar to what we did in mitosis. Except for now, when we get done, we're going to have two cells, but they're going to be haploid. But the problem is we still have our sisters. And so with our sisters, we've got to get rid of those. So here was our meiosis one. So we had our S phase, we made our sisters. We had in prophase one, we had our potential crossing over. We're slowly lining up in prometaphase. We have lined up in the middle in metaphase. We have our divorce here. We split those parents up, and now we're going to have two cells after we get done with cytokinesis, but they're still going to have the sisters. But we have a way of separating our sisters, and that is the same thing as what we did in mitosis. So we're going to go into meiosis 2. So meiosis 2, we're going to go right into prophase 2 and prometaphase 2. We are going to condense. If we unwrapped our chromosomes any, we're going to condense them. We're going to get rid of that nuclear envelope. We are then going to slowly line up single file as we prepare to go into metaphase 2. Metaphase 2, we're going to line up and we're going to split those sisters. So first we split our parents, and now we're splitting up the kids. All right, so as we split up those sisters, they're going to move to opposite ends of the cell for our anaphase 2. And then we're going to go into telophase 2 and cytokinesis 2, where we're going to separate those two cells and we're going to reform our nuclear envelopes. So now when we get done, we'll have 4 haploid cells. All right, so again, prophase 2, we're going to wind up our DNA and dissolve our nuclear envelope. Prometaphase, we're going to start hooking up those spindle fibers and pulling those chromosomes into their proper location, which is in single file across the metaphase plate for metaphase 2. Anaphase 2, we're going to separate our sisters. And then in telekinesis, wow, we're with our mental abilities. In telophase and cytokinesis, we're going to split those cells up into 4 genetically unique haploid cells. And so when we compare meiosis 1 and meiosis 2, so again, when we're looking at those, remember in meiosis 1, we split, this is the divorce. We're splitting our parents. In meiosis 2, we're splitting up the kids, the sisters. Split the sisters. So if we compare our meiosis and mitosis, we can see that in meiosis, there are 2 sets of cell divisions. And in mitosis, there's only 1. We still are going through our basic PMAT, right? So we have our prophase, metaphase, telophase, anaphase, and telophase, or as we're doing it, because we decided to throw prometaphase in here in the last couple of years, our PPMAT. All right, so we have our prophase, prometaphase, but we can see the process we still are going to go through. We're still going to go through our DNA synthesis. Both of them have that. They go through that cell cycle. They're going to go through the S phase. We're going to have synapses of the homologous chromosomes during prophase 1. We do not do this in mitosis. We will have a crossover in prophase 1. Again, we do not do that in mitosis. And then we have the homologous chromosomes line up. That's in metaphase 1. Do not do that in mitosis. Sister chromatids line up in the metaphase plate. That happens during metaphase 2. That happens just during regular metaphase and mitosis. And the outcome, again, we have 4 haploid cells at the end of meiosis 2. And again, they are genetically unique. They're very different. And some of them may not be very different depending on how they go. As you know, some siblings look very similar and other siblings do not look anything alike. But at the end of mitosis, you have 2 identical diploid cells. Now, there can be some errors, mistakes in meiosis. And with those mistakes, we can see them on what's called a karyotype. A karyotype is where we can get your chromosomes. We can take a picture of them. They don't line up nicely like this for us. We have to kind of arrange them. In this case, they used a fluorescent dye, and you can see how they have banded this. The bandings are similar, so we can match up our homologous chromosome. A karyogram is the chart. This is a karyogram or a picture of our chromosomes, and we can use this to identify genetic disorders. Now, karyotypes can only find gross genetic mistakes. They can't tell like if a gene is messed up, but we can tell if a chromosome is messed up. And in this case, we can tell that this individual would be a female, has two X chromosomes. Right there. Now, sometimes we have this problem called non-disjunction, and with non-disjunction, we can have a mistake in two places. We can have a mistake in anaphase 1 or anaphase 2. So when we go to do our divorce here in meiosis 1, notice we didn't split our parents. And so now these two gametes have an extra chromosome, and these two gametes are missing a chromosome. That can be a problem. Now here we have our divorce. Our divorce goes fine. We split our parents, but the sisters just didn't separate. And so they didn't separate right here. We can see they stayed together. That's a problem. So now this cell has one too many. This has not enough, but the bottom cell, it went fine. So we have the proper number of chromosomes with non-disjunction happening in the second cell division. We have at least 50% of the two of the cells. And I don't want to say 50%. It's not like all the eggs and sperm. This just happens in the creation. I don't even say the word creation, in the formation of one egg, one sperm. Not all, just that one had this problem. So we call euploid a human with 22 pairs of autosomes and one pair of allosomes. And aneuploid is where you are missing or have too many, one too many, or you can have more than one, but generally they don't survive to birth. But when we talk about monosomy, they're missing one chromosome. An example of monosomy would be Turner syndrome. And we're going to talk about all these in lab. Trisomy would have an extra chromosome. And so, and a good example of that is with Down syndrome. We can see that as females, when we form our eggs, let's see if we go into that. No, we do not. Females, when they make their eggs, oogenesis, we'll talk about this in lab. Their eggs sit in prophase 1 until they are ready to ovulate that egg. So your eggs have been formed since before you were born. So whether you're going to be a mom at 25 or 45, your expiration date on your eggs is definitely, time is ticking. The longer those eggs sit, the more difficult sometimes it is for those parents. Those homologous chromosomes don't want to separate. And this is what leads to older parents generally being a high-risk pregnancy. I should say older moms, because dads, you recycle your sperm constantly. It's like something like every 72 hours, you're putting a fresh stock of sperm out there. You break down your old, you make your new. But us women, nope, they're getting old, they age with us. And so we can see there's definitely a correlation with the age of the mom and the frequency in which babies are born with Down syndrome. Polyploidy, more than the correct number of chromosome pairs, are rare. Polyploid organisms generally are sterile and cannot do meiosis. An example of in plants, it's a totally different story. In plants, polyploidy usually makes the flowers bigger, the fruit bigger, but in animals, they usually don't survive. And so we see that exception in plants. We also can have, during crossing over, we can have mistakes that happen. We can duplicate, we can lose genes, we can delete genes. Sometimes we can get duplication of genes. And so we can get a couple of things that happen during that process. We can also get an inversion where they get flipped, or we can get a translocation. A translocation would be where you have a crossover with basically a cheating chromosome. So when those homologous chromosomes pair up, if they don't pair up properly and they've paired up with not their partner and they switch DNA, that would be a translocation. So that's a crossover with a non-homologous chromosome. And sometimes these issues may not cause that individual any problems, but it may cause problems with them having children later in life it could result in them being, I don't want to say they're sterile. Sterile generally implies either low sperm count or your egg count is low. They have them, but because their chromosomes are different, they have a hard time when the egg and sperm come together to fertilize, because one of the things they do is pair up their chromosomes. Homologous chromosomes pair up before they undergo that first mitosis. They make sure everything's in check. But if they can't recognize their partner, they can't get it together. And so mitosis fails to happen. So the embryo does not form. So those types of chromosomal structural rearrangements, like I said, it may not cause that individual any problems because all the DNA may be there, all the genes may be there, but it may cause problems later with having children. All right, so that concludes this chapter. Hopefully this lecture was helpful. And like I said, if you have any questions, we'll be talking about a lot of this stuff again in lab. All right, I will see you in the next video.